Hi, today we're going to have a look at excretion, or at least we're going to start having a look at excretion. Excretion is an example of a homeostatically controlled process. Excretion involves lots of different things. Breathing out carbon dioxide is an example of excretion. Quite often pooing is wrongly assumed to be excretion, and we're going to have a look at that as well. So we're going to have a look at the process of excretion. It involves two different organs. It involves the liver and then the kidney. The liver makes the excretory products that we're going to talk about, and then the kidney gets rid of them. So we're going to move on to the kidney after we've had a look at the liver. So today we're going to focus on the liver itself. We're going to have a look at the structure of the liver, and we're going to have a look at its functions, and then next lesson we're going to have a look at one of those functions in much more detail, which is the production of urea. So that's going to be next lesson. So for today we're going to have a look at excretion, and we're going to have a look at the structure of the liver. So what is excretion? Well, strictly speaking, excretion is removing waste from metabolism, so from metabolic processes. So getting rid of carbon dioxide, breathing out carbon dioxide, is excretion because that carbon dioxide was produced during respiration. Getting rid of urea is excretion because that urea was produced through a metabolic process which was breaking down excess amino acids. Pooing, strictly speaking, does have some excretory products. It has something called bilirubin. Bilirubin is a breakdown product of red blood cells, or more specifically hemoglobin. So when your red blood cells come to the end of their life, they're broken down by specialist cells called Kupffer cells, and bilirubin is produced when you break down the hemoglobin, and it is a pigment. There are lots of different kind of bilirubins and urobilins, and they are what makes your wee yellow. So urobilins make your wee yellow, and bilirubins make your poo actually a brown colour. And if you're born jaundiced, that is some of those products of the breakdown of red blood cells, because when the baby's inside the mother in the uterus, you can't just kind of wee out all of those things. And so sometimes the mother's body can't quite cope with the volume of these pigments that are made, and so they can deposit in the fatty tissue underneath the skin of the baby, and that's what causes them to be jaundiced. And quite often when they're born, they have to go onto those UV beds. I don't know if you've ever seen a baby on a kind of a blue light bed. It's actually UV light. They're sometimes called billy beds or UV beds, and they break down these pigments, and then the baby just gets rid of them when it wees as normal. So anyway, so we're going to have a look at these excretory products. So we're going to talk about bilirubin, we're going to talk about carbon dioxide very, very briefly, and we're going to talk mainly about the urea, and we're going to look at the urea much more closely next lesson. So pooing is getting rid of the things that you can't digest. So the fibre in fruits and vegetables, whole grains, you can't do anything with that. You can't break it down because you don't have the enzymes required to break it down. So you just poo it out. Now, you might think that's a little bit wasteful, but actually it keeps your bowel healthy because it keeps things moving along. The fewer of those fibrous materials you eat, the more that kind of stuff stays in your large intestine, and it can lead to problems later down the road. So it's good to keep things moving, and that's what the fibre does for us, but we can't actually digest it, so that's why it's not excretion. We haven't done anything to it. It is not a waste product of metabolism. It's just something we need to get rid of because we can't do anything about it. Right, so what is excreted? How are they formed? So have a little think. What processes formed the things that I've talked about? So carbon dioxide, you should remember, is formed during respiration. Bile pigments, they're the things that are formed during the breakdown of hemoglobin when red blood cells come to the end of their natural life. Red blood cells only hang around for about 3 months. They don't have a nucleus, so they can't hang around for very long, certainly not as long as some of your other cells. But it would be wasteful just to get rid of all of that hemoglobin. So we break those pigments down, we reuse quite a lot of the bits, and then the bits that we can't reuse, They're excreted as bile pigments. So they are actually excreted via the kidney. So they're excreted kind of, well, actually they're excreted in your poo, sorry. So bile pigments are quite often excreted in poo. Now, that actually has its own proper term. I'll come on to that in a minute. Anyway, carbon dioxide, we breathe that out, obviously. So that's breathed out in the lungs. Obviously, we poo that the bile pigments out. And then the nitrogenous waste, the urea, that is broken down. So excess amino acids are broken down in the liver, and then that nitrogenous waste broken in liver, sorry, and then excreted, this is terrible, in the kidney. That's terrible, sorry. So carbon dioxide produced during respiration, breathed out in the lungs. Bile pigments produced when we break down hemoglobin and red blood cells, excreted in the poo, so via the large intestine. Nitrogenous waste is what happens when we have excess amino acids. That's broken down in the liver and then excreted in the urine from the kidney. So the kidney is what actually gets rid of the urea, and it's excreted in the urine. Now, the term for pooing is actually something called egestion. It is not excretion, it is egestion. Now, obviously getting rid of the bile is excretion, but generally speaking, we call pooing egestion. So where is the liver and what's it kind of linked into? Now, the liver is large. It's about 5% of your body mass. And if you put your hands on where you think your stomach is, you're actually putting your hands across your stomach and your liver. Your liver kind of curves off to the right hand side and your stomach kind of curves up here on the left hand side. Now your liver actually partially obscures your stomach. Your stomach would be kind of, these two things would be a bit more closely associated. So your liver's right at the front. And if you put your hands on, just under your boobs, or at least they're just under mine because I'm a little bit old, and it feels warm. It feels very warm. Your liver is highly metabolically active. It's the most metabolically active organ in your body, and it generates an awful lot of heat. And so that's why if you put your hands there, it feels very, very warm. I've got another picture on the next slide. We can have a closer look at that as well. Now, your stomach and your digestive system will obviously break down your food. So it'll break fats down into glycerol and fatty acids. It'll break carbohydrates down into glucose, and it'll break proteins down into individual amino acids. They will all be absorbed into your blood. Now, along with that, you could potentially absorb toxins. So all of those digestive products, what they do is they go to the liver first. And so you can see, where is it on here? You can see this blue one here. It's what this is pointing to. It's something called the hepatic portal vein, or it sometimes gets abbreviated to HPV, which lots of other things get abbreviated to HPV. Anyway, so your hepatic portal vein links your digestive system to your liver. So all of those products of digestion and any potential toxins are taken first to your liver. So if you have excess glucose, it is stored in your liver. If you have excess amino acids, then they are broken down in your liver and it forms urea. If you have any toxins that you have digested or absorbed, then they're taken to the liver and your liver detoxifies them. So that is one source of blood going into the liver. So your hepatic portal vein goes in to your liver. Okay. Now, as with every organ, you have to have blood going in, and that is the hepatic artery. So if it's an artery, it's flowing away from your heart. So if it's going away from your heart, it's going to the organ. So it goes into the liver. And then you've got the hepatic vein, which is taking blood away from the liver because it is going in to the heart. So veins go into the heart, so it must be going away from the organ. So you've got the usual setup, an artery going into the organ, a vein going away from the organ. So the artery is taking oxygen-rich blood into that organ, and the vein is taking carbon dioxide-rich blood away, and any toxins away, any kind of carbon dioxide, sorry, I should say. Obviously, any toxins are broken down in your liver. So oxygen-rich blood is going in, carbon dioxide-rich blood is coming away. Okay? And then on top of that, you've got your hepatic portal vein. Now, it's called a vein, it has the structure of a vein. It's not coming from the heart, it's not under pressure, so it is just a vein, but it links your digestive system to your liver. Okay? And this is where students go wrong, because you have two vessels going in to your liver. You've got the hepatic portal vein and the artery, but you still only have one artery going in. Okay? Right, so this one shows, so if you were to draw the stomach on here, it would sit under there-ish, somewhere. Right, so you can see here you've got your hepatic artery and you've got your hepatic vein. They're carrying blood, the artery is carrying oxygen-rich blood in to the liver. The vein is taking carbon dioxide-rich blood away from the liver, and it's also going to be taking urea away from the liver because the liver forms urea. And then you've got your hepatic portal vein, which links your digestive system with your liver. Okay, so you can see this absorbing all of those products of digestion and then taking them into the liver. Okay, so I think I said most of this-ish. So your liver, it's large, it is reddish brown. Now obviously if you see liver in the shops, that's quite a dark brown, it's quite a dull brown. It would look a bit more red in life, or if it was fresh, because obviously you'd have fresh blood going through it. But it is a browny reddy colour. It is large, as I said, so about approximately 5% of your body mass, and it lies just underneath your diaphragm. It has several lobes. Now I'm going to talk, I think on the next slide, about lobules. Lobes are, your lungs have lobes. It's the kind of chunks of lung, chunks of liver. Lobules are smaller units. So we're going to have a look inside the liver, and the liver is made up of many lobules. But on the outside, it looks like it kind of comes in parts, and they are the lobes. So you can see kind of like lines down it, sort of chunks, I guess. It's fast growing and it can regenerate. You can reduce your liver to about 1/3 and it will regrow. And so if you are unfortunate enough to have any kind of trauma or sort of cancer to your liver, you can have a lot of it removed and it will regrow and you will hopefully survive. It has a very rich blood supply because it has two blood supplies. It has the hepatic artery and the hepatic portal vein. So it has the products of digestion. So it is rich in glucose, amino acids, glycerol, fatty acids, and it also has the hepatic artery, which is rich in oxygen. So it has this really rich blood supply going in. Approximately 1 decimeter cubed flows through your liver per minute. Now, for those of you who don't know, a decimeter cubed is the same as a litre. So if you go and have a look at a litre of milk, that's how much flows through your liver per minute, every day, all day. And like I said, it's very metabolic active, so therefore it's going to have lots of mitochondria and Golgi. Mitochondria, because obviously it's very active, so it needs lots of ATP from respiration. Golgi and also ribosomes for making all of those enzymes. Your liver makes an awful lot of your enzymes, and so they are proteins. They're going to need ribosomes, and then obviously the Golgi, ER, whatever, to package those proteins and those enzymes. Right, so the functions of the liver. We're going to talk a little bit today about the production of bile. Now, obviously, if you eat more glucose than you can deal with, then you can convert that glucose into glycogen. So plants, if they have excess glucose, they convert it into starch. If mammals have excess glucose, they convert it into glycogen. Glycogen is stored in either muscle or your liver. And I don't know why I keep forgetting the I in liver. So muscle or liver, you store this glucose as glycogen. Detoxification of poisons. So like I say, if you ingest something, you have kind of no idea what's in your food really sometimes. Then your liver will detoxify those poisons. And then deamination. Deamination is getting rid of the amine group on a protein. So we did this at GCSE. We talked about, I mean, obviously you know the formation of an amino acid. So you've got your R group, you've got hydrogen there, and you've got your amine group here. Deamination is taking that off. This is all incredibly useful. Carbons, hydrogens and oxygens, you can make everything else you need from those. They're the kind of the base units of all of your biological molecules. This is trickier. We only really use it in amino acids. So we'll have a look at this more closely next lesson, but that's what deamination is. Deamination is this, and it forms urea. So if you take two of those, you get urea. So two of these amine groups will form urea. So that's what we mean by deamination of nitrogenous waste. Nitrogen containing waste, that's what nitrogenous waste is. So we'll just get rid of this for a second. So this nitrogenous waste means nitrogen containing, and you can see it did have a nitrogen there. Okay, so that's just to recap those blood vessels. It is really, really, really important that you remember that. So the two that are going in to your liver are the hepatic artery and the hepatic portal vein. They go in, and then your hepatic vein leaves. Okay, so oxygen-rich blood in, deoxygenated blood out, products of digestion from your digestive system to your liver in the hepatic portal vein. Right, so what does your liver look like inside? It is very uniform in structure. The individual cells are called hepatocytes. So hepatocytes are liver cells. Now, you can call them liver cells in an exam. You will still get the mark. Hepatocytes is the more technical term. Anything that is hep or hepato is liver, hepatic portal vein, hepatic artery, hepatic vein. Hepatocytes, cytes means cell. Cytolysis is the lysis of cells. Cytoplasm is the watery bit in cells. So hepatocytes are liver cells. Now, here we can see one of those lobules. So this, if you looked at kind of at a more lower magnification, you would see the liver is made up of lots and lots and lots of these, hundreds if not thousands of these. These are the lobules. So these are small. You can't see them with the naked eye. So the lobes are the kind of the parts of the liver that you can see just looking at the liver. The lobules you can only see with a microscope. So your liver is made up of lots and lots and lots of these. Now those hepatic portal vein, hepatic artery and hepatic vein that I talked about, they come up here between the lobules. So they come up in these gaps between the lobules, and then blood will flow across past all of these cells, and then once the kind of the liver cells, the hepatocytes are kind of finished with the blood, it all drains into this, which is called the central vein. Now the central vein will start to kind of collect that blood, and eventually the central vein will come together to form the hepatic vein. So that is a branch off the hepatic vein. That's terrible writing, I'm very sorry. So the central vein, you've got many, many central veins. You've got one inside each of these lobules. They come together to form the hepatic vein, and the hepatic vein takes that deoxygenated blood away from the liver, and that deoxygenated blood is going to be rich in urea, because one of the jobs, one of the functions of these cells is the formation of urea. Okay, so that is just one lobule. Can I get rid of that a little bit? Okay. just to recap, you can see here you've got one lobule. So one lobule would be this thing. And you can see you've got many of them. You just kind of keep repeating. Now we quite often draw them as hexagons. As you could see from the last slide, they are not hexagons. They're not that uniform, but they are still quite uniform. So we do tend to draw them as hexagons. So you can see each one of them has a central vein here. And the blood is flowing in towards that central vein every time. It flows past and it's going in to that central vein. Okay, and you can see those blood vessels in the kind of the corners, so in between the lobules. So you've got the hepatic portal vein and the hepatic artery both coming up here. Let me see if I can make that a little bit thicker. So the hepatic vein and the hepatic artery blood goes up, and then the blood mixes and it flows past these cells and eventually exits into the central vein. Now, and you'll see the hepatic, so these central veins all kind of come together to make the hepatic vein, and that takes everything away again. There is another vessel in this little collection of vessels, and you can see it over here. It is the bile duct, and you can see it here as well. It is the bile duct. So in these corners, you've got the hepatic artery, the hepatic portal vein, and the bile duct, and you need to be able to identify which one is which. So hopefully you can remember that the hepatic portal vein has a much larger lumen than the artery. The lumen compared to the thicknesses of walls is much greater in a vein compared to an artery. So the hepatic portal vein will always look like the biggest one. It will always have the biggest lumen in these corner spaces. The hepatic artery will kind of look a little bit, remember when we did animal transport, we looked at arteries, and the walls of them are thick and kind of muscular, and they kind of look a little bit stripy. The bile duct, it is a duct. It is made and lined of cells. So you will see individual cells making up the bile duct. And inside each of those cells, you will get a nucleus. Can't really draw a nuclei in those. So you'll be able to see the difference between these kind of layers of the hepatic artery and then these cells of the bile duct. Okay, so you need to bear that in mind, because they have given these diagrams for you to label before. Now you have space in your book to draw a liver lobule, and so I would draw this. Okay, so if I just take all of this back off so you can see, then you are going to be drawing that, and you need to label the hepatic artery, the hepatic portal vein, the bile duct, and the central vein. And remember, the central veins all come together to make the hepatic vein. You might want to just add that as a little note at the bottom. Okay, so let's have a look at some pictures in a minute, obviously. Right, so this is just a portion of one lobule. So you can see they've just taken a part of the lobule here. So this is the central vein. So this is the central vein. And as it says there, a branch of the hepatic vein. So eventually they'll come together to form the hepatic vein. You've got your hepatic portal vein and you've got your hepatic artery. Now the blood comes up through these vessels, and then it mixes as it flows past these hepatocytes, and then eventually will drain into the hepatic portal, into the hepatic vein. But these two bloods come together and flow through things called sinusoids, and you can see there. So these sinusoids are channels, and they run past all of these hepatocytes. Now these hepatocytes all have microvilli to increase the surface area. You want lots of exchange. You want oxygen going into the cells. You want the amino acids going into the cells, the glucose going into the cells. You want urea coming out of the cells. You want carbon dioxide coming out of the cells. And so you want those waste products, the urea, the carbon dioxide, coming out into the blood and then it can drain and be taken away. Okay, so you've got blood coming up through the hepatic portal vein and the hepatic artery, and they mix as they flow through these sinusoids, past all of these cells with microvilli, plenty of surface area for exchange, plenty of mitochondria, plenty of activity going on. Now you can see here, if I can highlight this special thing here is called a Kupffer cell. And Kupffer cells are specialised macrophages, and what they do is that they do that whole breaking down of those red blood cells that have come to the end of their natural life, and so that's where you get the production of the bilirubin. So those red blood cells, like I said, last about 3 months-ish, and then they're broken down, and they're broken down by those Kupffer cells. So that's the kind of deamination element. The deamination involves the sinusoids, the hepatic portal vein, and the hepatic artery, and a little bit of the Kupffer cell. Now you can see over here, you've got this separate little thing going on here, and this is bile production. So let me just get rid of all of that again. So this is bile production. So you can see here we've got a branch of the bile duct. Now you've still got hepatocytes, and you can see them lining this channel here, lining this channel. Now this channel is called a canaliculus here. Now the hepatocytes lining that channel, lining this canaliculus, produce bile. And so they are kind of taking these breakdown products of these red blood cells, getting this bilirubin, getting these bile pigments, producing this bile, and the bile flows in the opposite direction to the blood. So it's flowing this way, it's flowing away from the central vein, and it flows towards the bile duct, and then will be taken away. So you can see at this end, if I can highlight that a little bit more, it's not open to the central vein. So the bile cannot get into the central vein. It flows in the opposite direction, so it's flowing that way, where the blood is flowing that way. Okay? All right, so let's have a look at that in words. Oh, another diagram. I did look at this slide. It shows you the same thing. So you've got the hepatic artery, the hepatic portal vein, that blood flowing past these hepatocytes into the central vein, and then this is the bile flowing in the opposite direction, and then that will be taken to the gallbladder, and eventually that will be excreted in your digestion, in your poo. Right, so here's some nice micrographs here, some views down down microscope. So you can see the portal vein there, much larger lumen. The vein has a much larger lumen than the artery, and you can see the artery and the bile duct very, very, very similar sizes, very similar sized lumens, but the bile duct is lined with cells. That's what you're looking for. Many, many, many nuclei. It's much clearer on the next slide, if that is on the next slide. And then you can see in the middle of that lobule, you've got that central vein. So that's what that is. That's the central vein right at the middle. So here you can see a very nice picture. You've got your hepatic portal vein, really nice big lumen, and you can see the difference here between your bile duct and your hepatic artery very nicely. You can see those layers of the hepatic artery, whereas you can see the individual nuclei making up the cells that line that bile duct. Let me just get rid of that so you can see it. So you can see individual nuclei. See if I can draw that. So you can see these nuclei. Oh, no, I want to go back. Oh, anyway, go back and have another look at that one. So let's have a look at the functions of the liver. So blood enters the liver from the hepatic portal vein and the hepatic artery. The two mix as they flow through those sinusoids, past those hepatocytes. Those hepatocytes, many microvilli, really large surface area. Oxygen, digestive products into those hepatocytes, get a good exchange between those things. Blood exits into the central vein drains into the hepatic vein. Kupffer cells break down the old red blood cells, you get that formation of bilirubin, and that's excreted in the feces, and that's what gives you your feces, your brown colour. So if you have a high turnover of red blood cells. It kind of goes, it should be a nice healthy chestnut colour apparently. There you go. Right, so bile formation, it's produced in those hepatocytes, passes into that channel, that canaliculus, drains into the bile duct, stored in the gallbladder, and then it's taken away. So you have a gap fill, you've got some questions. If you have any questions about this, please pop them onto the Q&A sheet. I'll try to check that regularly and I'll answer any questions that you have. I will post on the Google Classroom what your homework is, but that's it for this particular lesson. Enjoy. Oh, look, another slide. Honestly, I looked at this. Have a go, see if you can work these out. So if you just want to pause for a minute, see if you can work out what 1, 2, 8 is. I am so sorry for my dog in the background. So 1 is that Kupffer cell, that's that macrophage that breaks down the red blood cells. 2, they are your hepatocytes. 3, that's your sinusoid, that's that channel that's got your blood flowing past those hepatocytes. 4, that's your hepatic artery. They've nicely colour coded it on this. Your artery is red and your vein is blue. It won't be like that on an exam. Your branch of your hepatic portal vein there, your bile duct and your canaliculus, and then your central vein or branch of hepatic vein. You can use either term in an exam.